Sorry for the delay. So welcome to, I think this is our last lab for these sessions, yeah? So basically we're going to learn, what have you learned from last week? You've learned on how to count how many bacteria there are in water, yeah? So you have spread this, and then today, this is from your spread plate, so it's actually very nice. Can you get one each? Like all of you. Can you pass to the back, please? Pass quickly. Yes, pass to everyone. Is that yours? No, for this one. Okay, pass. Make sure everyone has one. So make sure everyone has one. Okay, and the rest pass it to the back, please. Okay. Pusing-pusingkan dia. Nampak tak muka?
Nampak.
Okay. So if you see this basically, okay. Okay, so as you learned from last week, one single colony is going to grow and become a colony. From one single colony, you cannot see them. When they become a colony, you can see them and you can count them. Okay? So you understand about the serial dilution. But since your count was too little, basically there is no need to do serial dilution because your water was quite clean. Because you can only count between 30 to 300 on a plate because this plate is not very big. Guys, please listen, yeah? So instead of doing serial dilution, what you should do last week was actually to concentrate it. So you can use filter and then concentrate it more, but we didn't do it in our lab. Okay? So once it grows, you want to know what are the bacteria that you managed to get. Okay? So first, if you look at your samples, because yours is not very concentrated, like you see, like can you see single colony? Like one round. Okay. So maybe there is one colony from there, assuming it's actually one colony, but sometimes they are like too close to each other and you cannot split them. That is why, guys listen, Hazik kan namanya siapa? Hanif, okay, lain kali pula. Hanif. Zarif. Okay. So if it is a mixture of species, you need, before you know what they are, you need to isolate them. Basically, you make them single species. Because it might be a mixture. Some of them are mixture. Like this, for instance, if you see like that one, how are you going to split them? But you're lucky now because they are basically split. But if you leave this on to grow, it will combine. Okay. But then, if you want to identify them, you need to turn them into one single species. Okay. So what you can do is you can actually get that one. Because they might be like a mixture. Can you see like different color there? In your one? Yes. Okay. So if you want to know what they are, you need to isolate them into a single species. If it is single species, next time you can just grow and regrow them. It's always going to be the same. But if it is a mixture, like is that a mixture? Yeah, like this one, you see white, green. So if you grow them again, they're going to combine. Okay. So how you do it? How you do the isolation. So if you have this and then you make a new plate, you're going to do a technique called streaking. What streaking does is actually you take it and then you're gonna what is that thing? This one you're gonna take from one single sample and then you're gonna streak like this. And basically what it does is serial dilution, because when you streak towards that way, it's gonna be diluted until at the end nothing. Then you're going to streak again like this, you dilute it again, and like that, you dilute it again until the end, you're going to get single species. So basically, you're going to streak it like this. So this will be the most concentrated and become the most diluted. And then you're going to streak it again from here. This is the most concentrated, this is the most diluted, and you're going to streak it again from 2 to. So this is the most diluted. Okay, get it? At the end, you're going to get hopefully single species here. Like from single colony, it will grow, and then you're going to take this and put it into the next plate. You're going to get single colony. Because now we have, like most of you have multiple colony inside. So you want only one single colony. But actually, you are quite lucky. Like in this case, because they are they are actually quite, they are not combined, so you can just take them. But if they're combined, like they mix, then you cannot split them. So that's the technique we're going to learn today, which is to strip them. So we're going to use the things. Wah, Zarif. Dengar. Okay, we're going to use the sample from last week. Okay, so you already have a combination of bacteria. You're going to isolate them into single culture today. Okay, so that is the first step. Okay, that's what you're going to do today, yeah? But unfortunately, you can actually look at the results tomorrow because you need to wait until tomorrow before it goes. So all of you will be given one plate. You need to do your own streaking. I'm going to show you how to do the streaking. And then, actually, if you look at the lab manual, there is actually a link to the internet, the YouTube. You can actually learn from them. But I suppose none of you have seen it, can? I know. Okay, there is a link in the internet here. Okay, so that will be the first part. The second part is assuming that you already have this single colony. You're going to grow from here. So if you get a single colony at the end here, you can regrow from here, from the single colony, and you're going to get one single colony all the time. It's the same species, so it's no longer a mixture. You can know what species they are inside your sample. Understand? Okay. And then you're going to do gram staining. The next procedure is to know what bacteria they are. Gram staining can tell you about the cell wall. It can tell you whether the cell wall is thick or thin. Okay? And the next procedure is you can actually identify using DNA, which you're not going to do unless if you take forensics. Okay. So what gram staining does is actually you are going to put two different kind of what do you call dye. Okay, so gram staining, have you done gram staining before? No, okay. So gram staining can tell you the difference between thin and thick cell wall of the bacteria. How does it do it? You're going to use two different stain here. So you're going to have, the first one is crystal violet. What you will do is that you will put crystal violet into your bacteria cell. Okay? So crystal violet will actually penetrate, goes into the cell wall. Okay? And then you're going to wash it with alcohol. And no yet. You're going to wash the crystal violet first. And then you're going to put iodine. What's the function of iodine? Iodine is not to give color, but what iodine does is actually as a mordant or as a glue that will glue the. Oh, you didn't get here. Okay, so iodine will glue the crystal violet into the cell wall. Okay, and after that you're going to wash it with alcohol. Okay, what happens is that if the cell wall is thin, the alcohol is going to wash the crystal violet out. But if the cell wall is thick, the alcohol will not be able to go in and wash it out. Okay, so once you wash the color out, you're going to put safranin. So if the crystal violet is still there, safranin cannot go in. But if the crystal violet is already washed by alcohol, safranin can go in. Okay, safranin is red, crystal violet is blue. Okay? So if the cell wall is thick, what will the color be?
No, violet.
Yeah, it's blue. Safranin is red. I want to see if you understand. I'm going to repeat the process again, yeah? If you can get it. The question is, if the cell wall is thick, is it going to be blue or red? Okay, listen to the process. First, you put crystal violet. The color goes in, and then you put iodine to stick the color onto the cell wall. And then you're going to wash using alcohol. And then if the cell wall is thick, the alcohol will never be able to go in and wash the color out. If the cell wall is thin, then the alcohol can wash it out. Okay, and then you're going to put safranin. So if the crystal violet is still in there, you cannot put safranin cannot go in. If the crystal violet is already washed out, crystal violet can go in. So which one is going to be the thick cell wall, blue or red? You can discuss. I give you 2 minutes. Discuss dulu lah. Okay, so blue. Okay, so blue is going to be thick cell wall, which also means gram positive. gram positive is thick cell wall and gram negative is thin cell wall. So at the end of your experiment here, you are going to tell me whether the bacteria that you're standing is gram positive or gram negative. That will be your conclusion. I want to know. Okay, so far we do not know because where is it coming from? Your bacteria initially? Where is the water sample coming from? The pond down there. So do you think I know whether they're gram positive or gram negative? How can I know? I never stand there. Okay, so you're gonna tell me eventually. Okay. So that's all basically what we're gonna do today. Hi, Mariana. Hey, welcome Mariana. Okay, so the first thing for Ida, okay, I'm going to show you all how to do streaking first, okay? Can you all see? I think people behind, maybe you can, I don't know if you want to come forward and see, because this one, if you haven't Googled it, or if you haven't looked at the YouTube, but there is YouTube, on your link on your lab report, the lab manual, you can actually go back and look at the YouTube link. But if not, come here. One Ida, do we have extra plate-nya? Adanya? Boleh tak? Kita makan saja. Bukan sebab kita bukan spread lagi. Memang kita tak. Ini tak. Bagaimana kita tahu kan? Ada ekstra.
Ada. Ambil extra plate, tengok yang cantik sikit.
Okay, so if you see this, every one of you is a mixture of culture. So this is not a good example, but I think I saw some. This one. All of the exchange. If you see, there is a mixture of color: yellow, white, and so many. Yeah. So if you keep on growing this, it's going to be a combination of species. You do not want that. You want a single species. So if you say that this bacteria is going to be able to remove whatever pollutant is, you do not know, because they're going to be like a lot of them. You want all single species. So what you're going to do today, like we said, you're going to streak them, and then basically by streaking, you do serial dilution, dilute them, and then at the end, you're going to get single species here and take it out. Okay? But in reality, this is also almost like a single species. That's it from yesterday. Okay, to isolate it. Okay, so how do we do that? Thank you, Ponaida. We have this plate. We're gonna open it first. Okay, remember, the flame has always need to be like as strong as possible, because it's basically sterilizing the whole area here. So if the flame is weak, bacteria is gonna go. And of course, do not talk like the way I do, because the bacteria from my mouth is gonna go into here. So first thing you need to do is like spray the whole thing down there with alcohol, and then clean it like we did before. And then this one, if the flame is strong, you're gonna sterilize the whole area here. If we have laminar flow, you can also use laminar flow. But if you don't have, according to standard procedure, this flame alone is enough. Okay? And then you see, like I'm gonna isolate the yellow ones. Look at that yellow ones. So the first thing is you're gonna burn this, because this is going to kill all of your bacteria, yeah. Like last week you used the rod glass, yeah? Okay, but actually I want to show you still, because last week our alcohol didn't work, so I want to show you, mana rod one? Dah ambil dah? Mana? Sorry, sebab because last week kita tak boleh kan, so I want to show them. Yeah, ada tak? Sorry. We just want to show you because last week our hockey stick doesn't work, yeah? So we want to show, yeah. It is called hockey stick. Sorry, tukar topik dulu. But I just want to show you how to do hockey stick. Last week it didn't work because our alcohol was not right. This time, okay. Ini tak apalah, tukar nanti ya. This is not part of it. I just want to show what I remember because last week our hockey stick doesn't work. Bagus you all belakang. Nasib baik you all datang semua. Ada pun aja? Okay. Ada alkohol juga ya? Okey. Ini alkohol baru ya?
Saya gantikan dengan yang besar.
Ganti ya.
Tidak yang kecil.
Iya. Yes, thank you. So last week, before you all do spreading, the hockey stick is actually supposed to be like this, but it doesn't go last week, but it should be like this. Yes, like this. It didn't happen last week, kan? And then you wait until it dies, and then you put onto the air and spread. But it didn't happen last week, yeah? Because of alcohol, I think, because we keep the alcohol for so long, and the alcohol evaporate and water, there's a lot of water inside. So this is what we should have done last week for the spreading. Okay. Thank you, Puan Aida. Tepuk tangan dengan Puan Aida. But this week, streaking. So I already burnt this. Can I do it now? Cannot lah. I already leave it for a long time over there. So I need to burn it again until it's red. That's fine. Leave it until it's basically cold a bit. But it's gonna be warm, hot. But you're streaking now. Normally, what you do, you can also touch it first into the agar plate to cool it down. But because now we are isolating, so we don't care because it's actually there will be a lot of bacteria. So I'm gonna stop talking here. So I'm not gonna talk. So when it opens like this, no, because bacteria is gonna go in. So never do like this. The moment you do like this, basically your sample is wrong already. Like this. I'm gonna try again, burn it. So this is the right thing you do. I'm gonna watch you. No talking here. Look. I'm gonna touch one single species. And then I'm gonna do like that. Open it and then see the way. And then burn it again because you need to kill the bacteria again because you're doing serial dilution, basically dilution here. Okay. Once it's cold, put it there, the place where you don't have anything, and then very warm. And again. I didn't do very well, but hopefully you guys will get and then like that. So tomorrow hopefully you will get a single culture.